We are running out of storage devices. Humans are going to generate around 200 zettabytes of data every year by 2030. It's a stupidly big number. If you try to store that on floppy disks and line them up, they'd reach from Earth to the Sun and back, roughly 300 times. Where we are going to put all of that data? The Mother Nature solved this storage problem millions of years ago. Deoxyribonucleic acid, or DNA. It's the molecule that stores the blueprints for every protein in your body. The proteins that build your cell, carry oxygen in your blood, repair damage, and basically keep you alive. All of that is written in DNA. DNA is incredibly durable. We've read DNA from fossils after thousands of years, and it's insanely dense. A teaspoon of DNA could, in theory, store more data than today's largest data centers. So, if DNA is this good at storage, why aren't we using it as a hard drive? In this video, we're going to see how that might actually work. First, let's back to the basics. Alright, so imagine you actually wanted to turn a real file into DNA. For example, I want to save this image of Sir Isaac Newton within a DNA. How am I supposed to do that? First, we must know that this image as a digital file is nothing but a long sequence of zeros and ones called binary code. That's it. Every photo, every movie, every message you've seen is just binary codes. If we want to store this image on DNA, we have to translate its binary code into DNA code. So, what's DNA code? DNA sequence is composed of 4 letters: A for adenine, C for cytosine, G for guanine, and T for thymine. One very simple way to bridge between binary code and DNA code is this. 00 equals to, for example, A, 01 equals to C, 10 equals to G, and 11 equals to T. So you take your file, you turn it into a long binary string, then you read it 2 bits at a time, then translate it into DNA code. For example, say a tiny piece of that file is 01001110. You need to chop it like this. Now you apply the translation rule. 01 to C, 00 to A, 11 to T, 10 to G. So this 8-bit chunk becomes CATG. In other words, the exact Isaac Newton photo is now written in different alphabets. Later on, when we read the DNA back, we just need to flip the mapping, then put those bits together again, and you get the original binary code. This is the core idea of DNA data storage. It's not magic. We're just relabeling bits as genetic bases in a way we can reverse it. Clean and easy, right? But biology is not clean. It's not black and white. It's not an accurate engineer. If we just naively translate binaries to DNA sequence, we run into biological problem. In fact, the DNA synthesis machines within the cells and sequencing machines in genetic laboratories do not like certain patterns in the DNA sequence. The first problem is homopolymers. These are long stretches of a single letter, like this and this, and they cause read errors. The second one is what we call GC content or GC ratio. For example, if a given sequence has, say, 90% G and C, which we know they are paired together in DNA molecule, or on the other hand, when it has little amount of GC content, like 5%, those sequences are abnormal and can form undesired, or what's the better word, nasty secondary structures. It means in case of very high GC content or very low GC content, the DNA would fold on itself, which makes it hard to copy. The third problem is mutation. We know that when we synthesize DNA, when cells replicated, and when we sequence it back, some letters will be wrong, missing, or inserted by accident. So the DNA version of our code might not be that clear. Problem number 4 is DNA motifs. We know that certain patterns of DNA sequence can form secondary structures of DNA. Enzymes within the cells may recognize these structures, cut them, bind to them, or degenerate them. And basically, it would mess up with our safe codes. So, homopolymer, GC content, mutations, and motifs, these are 4 main problems. If we are not careful, our binary code becomes a DNA sequence that is hard to synthesize, error-prone to read, or just behaves badly inside a cell. So to make a reliable hard drive out of DNA molecule, we need to somehow engineer around biology. To face these problems, scientists use tools from coding theory, the same mathematics behind Wi-Fi, QR codes, and CDs, and apply them to DNA. Specifically, they use something called constrained coding. That means when we convert binary to ACGT, we don't allow just any sequence. We enforce rules. For example, if we tell the encoder machine to never let more than a few identical bases appear in a row, we don't have homopolymers anymore. Also, we can enforce rules to keep the GC content in a safe range. So before turning bits into DNA, we deliberately add redundant information. So the original data can be reconstructed even if some fragments are damaged. In fact, this is not just theory. Around 2012, researchers published landmark papers in journals like Science and Nature showing that you could actually do this in practice. Take real digital files, encode them into synthetic DNA, and then read them back with high accuracy. These early experiments were small in size, but they proved that with the right encoding rules, DNA could behave like a serious, if very weird, storage medium. Okay, let's sum up what we said up until now. Imagine your biologist friend walks in and says, "I've got $1 billion idea. If we print all of our data into DNA, seal it in a little tube, and come back in 100 years, they will be safe." Probably we will tell them, yes, it's beautiful on paper, but at the end of the day, you end up with nothing that looks like a hard drive, just some boring liquid in a few Falcon tubes. Sure, they quietly holding more data than a room full of servers, but aren't they just the laziest archives you've ever seen? They are surely a time capsule, but not a USB drive you could use every day. Then your biologist friend will reply, what if the DNA belongs to an alive organism? Well, they're right. Instead of writing DNA into a tube and freezing it, you sneak that DNA into something like bacteria. Bacteria are already good at one thing, which is making copies of themselves. You give them a little ring of DNA that encodes your data, and every time they divide, they copy that ring too. Suddenly, your archive isn't a stack of tapes in a vault. It's a colony, a living population where each cell carries a fragment of your information. On a good day, this looks like genius. Copying now becomes biology's problem instead of yours. Your archive can in principle scale itself by growing. On a bad day, you remember what biology actually is? Mutations, selection, accidents, evolution. That means your storage medium now has opinions. It reacts to the environment. It can die, sure, but on the other hand, it can also adapt. It can survive. What was Charles Darwin saying? Survival of the fittest? Therefore, we should be careful because it's dangerous. So basically, with technology that probably will appear soon, we are going to trade a cold, fragile hard drive with low capacity for a warm, unreliable life with much higher capacity. We have also another problem. This alive, carbon-based hard drive is no longer a physical device. It's a population of organisms carrying our data in their DNA. how we are going to use it on our silicon-based computers. Is that a good idea? Well, maybe only for very specific controlled cases today, but again, it opens up a question we couldn't even ask with silicon devices. If the storage medium is alive, can it write its own data? On a good scenario, maybe some biologists got together one day and create something like Carbon Valley, which would be way stronger than Silicon Valley in order to help the humanity. But in a bad scenario, we don't even know what apocalyptic era is waiting for us. Now, let's go on the edge of science. What we have done up to now is pushing data into DNA from the outside. Now I'm going to tell you how to flip it. What if cells themselves could use DNA as a kind of journal? Not a static archive, but a logbook. At this time, I saw this signal. At that time, I got affected. Here a gene was turned on, and seconds later, a gene was turned off. And surprisingly again, the mother of nature already has a prototype of this. In fact, bacteria use a system called CRISPR-Cas to remember the viruses they've seen before in order to destroy them. Pieces of viral DNA get slotted into the genome in order, like adding new entries to a notebook. Scientists looked at systems like this and thought, wait, this isn't just a defense mechanism, it's a natural molecular recorder. In fact, in a famous experiment published in Nature in 2017, a team took a short digital clip. It was a few frames of a running horse. They converted it into DNA and used CRISPR to write those sequences into genomes of living bacteria. Later, by sequencing those cells, they reconstructed the frames of the movie while the colony of bacteria is still alive. The colony had literally become living hard drives, recording information over time inside their own DNA. So basically, they hijacked the CRISPR-Cas system and redesigned it a bit and get something like a biological recorder. Not a camera, not a microphone, a system that writes into DNA a sequence of events that happen to a cell over time. Think about what that means. Instead of successfully storing a photo into DNA, we now have a memory written by the cell itself. That means that DNA stops being just a shelf where we put our books. It becomes the notebook that the living organism itself uses to write its own story. I understand it's gone too far from the title of this video, but just think about it. The idea of storing our data on DNA is just stupid compared to having a system within the cells for writing everything we want in real time. So, after all of this, how do we answer the original question? Can we use DNA as a hard drive? If by hard drive, you mean the thing in your laptop where you install games and delete files without thinking, then no, not now, not soon. But if hard drive means a place to keep information over decades or centuries, densely and with a chance of being readable after, then our current devices are junk. Then DNA starts to look less like a gimmick and more like a candidate for the deep memory of our species. Of course, for DNA to be more than just a sealed time capsule, we also need random access. It's a way to pull out a specific piece of information on demand, not just dump the whole archive and sort it later. That's called the random access problem. Can you reach into a huge soup of DNA molecules and selectively fish out just the file you want? Researchers are already working on this. In a 2018 paper in Nature Biotechnology, for example, a team demonstrated methods to label and retrieve particular DNA-encoded files from a much larger pool, a bit like asking a hard drive to jump to one specific block of data. It's still slow and experimental, but it shows that DNA storage might not only be dense and durable, it might also become something we can actually query, not just freeze and forget. When I was making this video, one question kept bothering me. We know that our cells contain DNA, about 3.5 billion base pairs long. But What if this DNA is actually a code that could be translated into binary, or maybe into some other cryptic language? And I'm not just asking about humans. Maybe a cat's DNA is a code. Maybe a tree's DNA is a code. Who knows? At the very least, it sounds like a pretty good idea for a sci-fi movie, isn't it? My name is Taimur, and this is my YouTube channel of science and other demons. If you like this, I'll be happy if you subscribe. Have a nice day.